Hello guys, welcome to Big Book. Today we are going to discuss the frequency of cell division. Here in this slide we will discuss what we are going to discuss further in this particular experiment. Here we are going to compare the cells of the plant with the animal cells. Here we are going to use the example of onion cells with the whitefish blastula cells. Here we are going to study the cell division, where we are going to have these cell divisions frozen on as the specimen on the slides, and it is going to be possible to watch a single cell progress through the stages of mitosis. So it doesn't mean that the cell is going to divide and we are going to see it, but it is frozen one where we are going to see the cell progress like the stages or the phases. Based on observations, we are going to determine the frequency of cell division. Okay, here how frequently the cells are going to divide out of the whole mass within that particular organ that we are going to study. And further we are going to construct a clock representing the division cycle. So here we are going to develop the clock in the sense a spherical graph which is going to show the division cycle, where we are going to compare it with the complete one cycle of mitosis. It is going to be shown in the table. We will record the number of cells in each stage of mitosis. So this is how we are going to unfold the experiment. Now material required are compound microscope, lens paper for the lenses within the compound microscope, and the prepared slides of onion root tip. You know how to prepare the slides of onion root tip with different staining methods, and even the prepared slides of whitefish blastula, the developmental stage of the animal or the whitefish, which is the blastula, which is going to give a good amount of the cell division and also a particular perfect studies which we can do within this whitefish blastula. And also onion root tip is one of the best organ which we are going to consider here for our studies. Now here preparing the slides for onion root tip and whitefish blastula are the lengthy processes, so this can be studied with the different other sources even online on the books. So we have not included as these are going to be the lengthy processes. So further we are also planning for these two, how to prepare these particular slides, that will also going to be uploaded hopefully within short time. Next slide. In this we are going to do the observing with the help of the equipment compound microscope. Here we are going to set up and place the onion root tip slide, and we are going to focus on low power, that is 40x power, and further we are going to move this slide in the field and centered on the root tip, and further the focus will be changed to 100x. So here first is to focus the particular specimen which is present, where it is present on this slide, and further we are going to magnify it, and further we are going to recenter so that the focused on the more square meristem cells can be seen, and further we are going to focus on 400x. Slowly we are going to move this slide and search for cells in each phase of mitosis. When we are going to find the cells in the stages, you are going to do the observational drawing of that cell, and further we are going to label the important structures of the cell within the mitosis stages. So like this we are going to do this particular observing of dividing cells both in onion root tip and also in the whitefish blastula slides. Next slide. Here we are going to continue the observation until we are going to identify all the phases, whether it is in onion root slide and whether it is in the whitefish blastula. For both, the procedure is the observing procedure is the same. And further, we are going to take the note of these drawings and labeling the parts. Now further in the next slide, so here this is a particular typical diagram of the onion root tip, where the onion root tip here it shows the dividing cells. This is the interface cell. These are the interface cell or the non-dividing cells. Here it is the telophasic cells, sorry, anaphase cells. Here it is anaphase, that is the early anaphase cell. So here early anaphase cell. Here you can see the position where you will find the particular dividing cells, whereas at the top the dividing of the cells are very less because the organ has shown the growth. Here also the dividing will takes place, but the frequency will be very less compared to the frequency which is present at the root tip. So here, from here we are going to take the observational points or the diagrams or whatever, but here the observation can be done, but here the what the expected results will not be so much is going to hold good here. So next is, this is the whitefish blastula cells. Here you can see we have taken 3 diagrams, 1, 2 and 3. Here in this diagram you can see the cells are showing, this is pre-anaphase, this is metaphase, this is prophase, this is again prophase, this is again metaphase. Like this, here also you can see prophase and prophase. Now here you can see so many cells which are dividing and they are in their mitotic stages, that is the M phase of cell cycle. Now here this is the slide of the blastula. You know, blastula is one stage of the development of the organism where the division of these cells will be very frequent, so the high frequency will be there compared to the grown organism or full organism. In the same manner you can see here also, you can see the interface, early prophase, anaphase, prophase, like this. Here also you can see anaphase, prophase, metaphase. So in these slides you will observe many number of the frequently dividing cells. So like this you are going to make the observations and you are going to make the note of the drawings and labeling it. Further, you are going to do the determining the frequency of cell division. In this you are going to count around 200 adjacent whitefish blastula cells. Here 200 is a count which we have considered. You can consider even less or even more also as per your wish. So you can calculate further with the help of that. So you have to consider a particular n number of the adjacent whitefish blastula cells and record whether the cells are in interphase or division phase. Now here interphase or division phase is nothing but interphase is non-dividing cells, division phase is prophase, metaphase, anaphase and telophase. So here interphase is one, the preparing cell, but it is not actively dividing there. But division phase is one where the division of the nuclear material is taking place. So further is we are going to record the number of cells in interphase and the number of cells that are actively dividing. Further, you are going to repeat the same procedure for the onion root cell. So here we have considered whitefish, so here we are going to consider it for the plant root, that is the onion root tip cells. So here for the both, you either for one you can consider 200 adjacent cells, for the other you can take different number also. So either you can consider both the organisms with the same number of cells also. So here you are going to make the calculation like number of cells dividing, that is what we have considered as the division phase, divided by total number of cells counted. So other than the number of cells dividing are the cells which are total number of cells counted. So out of that we are going to do the division of this both, and we are going to multiply it by 100. So whatever we are going to get, that is the percentage of dividing. So that is what the frequency of cell division. So here to the whole amount of the cells which are not dividing, whatever the cells are dividing is the frequency. So, and you are going to create a circle graph showing the percentage of cells in division phase and the percentage of cells in interphase. So you are going to label the diagrams appropriately. Now here we have taken the values. This is for onion root tip. This is for whitefish blastula. Here, as we have observed, we got 65 dividing cells, and number of cells in interphase or non-dividing is 200. So here again, number of cells actively dividing are 48. Number of cells in interphase or non-dividing is 200. You can understand that what the value we have taken here. Number of cells in interphase or non-dividing. The total number is 200, which are not non-dividing. These are the number which are dividing, which are separate from this actively dividing or separate from not dividing cells. In the sense, the overall total number are going to be present within the adjacent sites within the view area of your microscope slide. So here you are going to do the frequency calculation, that is actively dividing cells divided by the total number into 100. So we got as 32.5% of the frequency in the onion root tip cells. Here again 48 divided by 200 into 100 is equal to 24% of frequency of the dividing cells. Now we have depicted this in the graph, circular graph here and also here. You can see this is the portion of the actively dividing cells compared with not dividing. This is the portion of the actively dividing cells compared to the not dividing. Now further, so creating a cell division clock. Here in the high power magnification, we are going to locate 50 onion root cells that are dividing, and we are not including cells that are between divisions. Now here dividing in the sense, again the prophase, metaphase, anaphase and telophase. Not dividing in the sense or not between the divisions in the sense which are after telophase and before prophase. So further we are going to identify the phase of mitosis each cell is in, and we are going to record the number of cells in each phase. We are going to repeat the procedure for cells with the whitefish blastula, as we have taken two specimens here in consideration. So we are going to do the both counting. So here for both the plant and animal cells, we are going to calculate the percentage of cells that are in each of these four phases, that is prophase, metaphase, anaphase and telophase. For each cell type, we are going to construct a circular graph showing the percentage of cells in each phase of mitosis. So we are going to discuss in the further slide this one. We are going to label and title the further the graph. If it takes 16 hours to complete one cycle, remember this point, 16 hours to complete one cycle of mitosis for whitefish and 12 hours for onions. This is the constant number of hours to complete the mitosis cycle. We are going to determine the time spent in each phase for both of these organisms respectively with their dividing cell count. So here further we are going to include this information in the circular graphs. Here we have performed the experiment. So here we are creating a cell division clock for onion root tip dividing cells. So we are going to consider the onion root tip, where entire cell cycle takes about 12 hours, that is 720 minutes. Further, we are going to calculate the percentage of time spent in each phase by, here counting the total number of cells in each phase we are going to do, and we are going to record the mitotic phases, that is the prophase, metaphase, anaphase and telophase, and we are going to record the number of cells which are appearing to be in the each phase respectively. And further, we are going to divide it by the total number of the cells we have counted. Here how we are going to do this is, like, we are going to 16 divided by 50 number of the cells which are considered here. Now here we are going to do the division, that is the total cells counted, that is the n 50 is going to be, this is going to be considered as n, and we are going to divide this 16 by 50 and we are going to multiply it by 100. So we are going to get this 32 as the percent. Now here this is the, like this we are going to do all the calculations where we are going to get the percentage of cells in each phase of the mitosis of the particular observed area will be considered, that is the 32% of prophase, 42% of metaphase, 24% of the anaphase and 16% of the telophase. The same percentage is also be considered to be the percent of time spent. Now here this is the percent of the time spent which we are going to consider for the percentages of the phases. We are going to do the same kind of calculations even for the whitefish blastula cells for the counted number 50 and the number which we have observed each phase of prophase, metaphase, anaphase and telophase, where we are going to consider the percentage of the phases and also the percentage of the time spent here, and we are going to do the calculation for the estimate of time spent. Here also it is estimate of the estimate of time spent. Here the heading has been given wrong, I apologize for that. So the estimate of time spent will be calculated like we have constructed this formula, that is C is nothing but the number of cells in phase. This stands for C. Here we have not mentioned, here in this table we have mentioned it. Now here N is for the total number of the cells and H is for the hours, constant hours. For whitefish the constant hours are 16 and for onion root tip the constant hours are 12. So here 16 and 12 are going to have 960 minutes and 720 minutes as constant. So these are the estimate of time spent for this particular number of the cells within that stage. Each one is respective to its each stage here. Now further we have depicted these values in the graphs. Now here you can see this is estimate of time spent, this is not percent of time spent. Here you can see 230.4 prophase, further is metaphase, next is anaphase and next is telophase. Now again here this is the percent of time spent in onion root tip. Here it is the prophase 32%, metaphase 42%, anaphase 24%, telophase 16%. In the same manner for the estimate of time spent, you don't go with this heading where we have done little mistake here. Here it is like prophase 364.8 prophase, next is the metaphase, next is the anaphase, next is the telophase, and next is the percentage of time spent in whitefish. Prophase 38, metaphase 48, anaphase 20, telophase 36. So this is the metaphasic circular graph which we have constructed to depict the estimate of the time spent by each phase together to the total number of cells which we have counted for each phase. In the same manner here the percent of time spent In the same manner here also for whitefish. This is for onion root tip. This is for whitefish. So this is regarding the frequency of cell division, which is counting for creating a cell division clock and also determining the frequency of cell division. So I hope I have given quite little brief explanation of this particular experiment. If there are any mistakes or wrong explanations within this, you can put it across in the comment so that I can rectify this slide and also give the more information. And I hope I had been very loud and clear for you guys to put across all the information which is present in this particular experiment of frequency of cell division. And hopefully we are going to give more number of explanations on few more topics regularly in coming days.